Okay, good afternoon. Yep, nice to see that still a lot of people manage to make it here so late on a Monday. Last time we had a look, we played a bit with this R sheet where we took a sample data set from the Sura standard 3K sample that we had run through the Sura standard workflow on our first time. And then we did it doing a lot of this stuff in a pedestrian manner. And we sort of, I think we stopped somewhere in the middle. Also, it's now more than a week ago, so I have to remember how far we got. I had hoped to prepare a bit more for you on the weekend, but to be honest, I was a bit sick on the weekend, didn't get as far as I wanted. So I don't have a nice polished script this time, but we can clean it up later. And I thought I now, the R script that I have prepared, maybe I quickly put it on Moodle. Then those of you who want to read along or add some comments can download it and add this for the, and we'll add a few more things later. And I'll come then, and I stick to a few points to see where we go here. So let me take this thing here, quickly upload it to Moodle in case that's of use for somebody. So you might have noticed last time I had this course started with course web page, but the server on which I put this course web page is currently disassembled in my office because the power supply made a spark and killed the main board. So that might take a while to get this, until this link to course material works again. So until then, I just upload what we need here. Dateidarstellungen. Upload, browse, 24. That's this one here, open. And maybe I also try to upload rows. The email I used last time, sorry, I should have done this before, but today was a bit hectic. Unless the downloads, maybe it's here. No, it's not. Just give me a second. Yes. cd tilde/temp. No, with this here. Datei, speichern und zum Kurs. Ja. Downloads. Yeah. The stuff from last week is on this server that broke down. So somewhere, but yeah, part of it. But this is why I copied part over to the new stuff, and I hope I get the server repaired tomorrow in the next days, and then everything will be sort of in a more clarified thing again. Unless temp cd ef Zip. Here we have something. Okay. While this thing is uploading the main data set, that's our example for today, I tell you what the example set is about. It's data from the research group of Anna Martin Villalba at DKFZ. So they are doing, in this group, they're doing neurobiology. I might have mentioned this already last time, but in case it's not, let's tell the story again. So one of the main research topics of the Martin Villalba group is adult neurogenesis. So to quickly tell you the part of the story about it, because we will use this example data set quite a couple of times. So to quickly tell you a bit about the biology. Those of you who are not biologists might still remember, the same as I did until I learned that it's no longer true here, from high school textbooks, that the brain is an organ that stays as it is. We are born with all the neurons we'll ever have, and after birth, no neurons are ever built. That's what you could read in most textbooks until 20 years ago. It's no longer true. By now, we know that even in the adult brain, there is new neurons being formed, but it's still unclear whether this has any functional consequences, whether this is a vestige from simpler organisms that were able to produce neurons, and maybe one possibility would be that that today, that the complicated brains of mammals like humans simply are not able to produce new neurons in the adult brain, which then manage to sort of find their way to replace something in this intricate network. But it seems that they can. For example, there's been a couple of trials with mice, and now even the clinical trials where they injected neuronal stem cells to treat neurodegenerative disorders like Parkinson, and it was noticeable that this improved the symptoms. So it seems that if you bring new neurons in this established connection structure of a grown brain, these new neurons actually seem to be able to find their way and insert themselves into all this connectome of all these connected synapses in a manner which is actually beneficial for the whole organism, which I think is quite is remarkable, and that's and hence there is this hope that maybe it can improve neurodegenerative disorder or traumatic brain injuries like strokes or the like, if we were able to make use of this, or maybe even to stimulate the production of new neurons in the brain. And this is why a couple of research groups, including the Martin Villalba group, research this growth of neurons in an adult brain. And a quite much used model system for this is the subventricular zone of a mouse brain. So the ventricles, are these empty spaces which are filled with fluid in the brain. And in the brain of a mouse, at the side of the lateral ventricles, sort of in the wall, it was noticed that there seems to be neural stem cells which are continuously, but at a very low pace and low throughput, produce always a few neuroblasts, so immature neurons, which then move along the middle of the brain towards the front, the nose tip of the mouse, and there replace some interneurons in the olfactory bulb. And it was also noticed that in case of injury, if you, for example, simulate a stroke in the mouse, that when this area, also surrounding tissues, suddenly start producing more neurons, which then also migrate to the damaged areas. So that made people quite excited, and this is why there is interest in studying this process. And we are now studying the normal process as it happens in a healthy mouse, where we haven't simulated an injury or something like that. And to this end, they dissected a couple of mice, took out this part at the brain, this so-called subventricular zone, so the neural tissue right next to the ventricle, and in this did single-cell RNA-seq on these cells. Now, even there, the vast majority of cells which you will find there are adult, mature, old neurons and glia cells, But what we are interested in is these stem cells and these new neurons. Hence, what we use is a method to get them specifically, namely enrichment by by FACS. What does FACS stand for? Flow cytometry assisted cell sorting. So flow cytometry means you have your cells. We, as I mentioned last time, we need to find some way of changing our big blob of tissue into individual cells, which all individual are suspended in some fluid or in some watery solution. And we use antibodies to mark certain cells. Namely, what we know is that a specific protein called GLAST is present on astrocytes and on the stem cells and also on young neuroblasts, which have been recently produced by the stem cells. So we have an antibody which binds to that. The antibody is coupled with a green fluorophore or with some fluorescent protein, so that the FACS machine can excite the fluorescent protein with a laser and then notices that it's one of these cells which we are looking for, and then it applies a little electric field to move the water jet through which the cell goes, and so causes this cell to go into a special receptacle, and in this way we collect all the cells which have this desirable property, namely having this GLAST thing. So that's what they did. They dissected out brain tissues and they used this FACS machine in order to get cells which have this GLAST protein on their surface, and by this hopefully they got the stem cells plus the young neuroblasts and maybe neurons. And now we perform single cell sequencing here. This specific data set, because in this lab they made many such experiments and have quite a couple of different data sets. In this specific data set, they knocked out some receptor, and they also compared cells from young mice and from old mice, because they wanted to know how the dependence on this receptor changes with age. It turned out that the difference weren't that great. And so for our purpose, we won't even look at these differences, not for now, but just imagine it's all the same one thing. And we will see in a moment how these cells look like. I took this data set now because last time we used this peripheral blood, and it was a rather simple data set, as you might remember. We just saw these 3 clusters of cells, which we then identified as being the B cells, the T cells, and the monocytes. And this time the pattern will look a little more interesting, and so we can see a bit more. But just to remember where we stopped last time, we start with the same process as we did as we started last time. And again, I will not use Seurat, but I will do everything by hand, which is of course not how you would do it in practice. In practice, we have a Seurat tool. It's nice and it does all our work. And it has its standard workflow, but it's hard for us to see what's happening inside. So we do it with sort of pedestrian way of stepping through everything step by step so that we can more easily see what's going on. So let's see if I can make this here a little bit bigger, because I think this is zoom in. Is this big enough for everybody? Okay. So let me delete the thing to start from scratch again, where I started last time. So that's what I've now prepared for the last 2 hours. I hope it still works, because it was all done a bit quickly. So I again use these 2 libraries, the matrix and sparse matrix stats, because we work with these sparse matrices. And these are the 3 files that I've just tried to upload to R, and just to have a look at them again. This is how they look like. The first one contains all our counts, and it describes a sparse matrix. So you read this as in the matrix, in the first row, in column 9, 5, 11, there's a 1. And in column 9, 5, 14, there's a 8. And everything that's not listed is a 0. And then what we also have is these 2 other files, which give us the meaning of the rows of a gene, which tells us for each row of a matrix which gene it was, and which tells us for each column of a matrix which cell it was. And this time we not only have the raw information about the cell, about its barcode, but we have already some preliminary analysis results of what my colleagues have already decided what kind of cell this was. But for now, we will pretend that this work hasn't been done yet, and we'll just start from scratch and only take this. In R, there is this sparse matrix function, which takes these vectors and assembles the sparse matrix as we've already done, but I now use the one from the Sera package so that I don't have to assemble it manually. The matrix is quite big, so this takes a while, especially as I work on my small laptop, because the big server, as I said, is broken. That should still work for a bit. Before, I think it took 20 seconds or so. Should be done any moment. And then, as you remember, we have each matrix, each column in this matrix describes the read counts for one cell, and we wanted to normalize this. So here's how it looks like. Each row a gene, each cell, each column a cell. And if I add up the columns of this matrix, then I see for each cell how many reads have been found in total for this cell. So you see this time the experiment worked a bit better than last time. Last time we had around 2,000 counts per cell. Now we have around 10,000 typically. And I divide each column of I divide each value on the matrix by its column sum to turn the matrix into a matrix of fractions. That means it tells us how many of all the reads for a given cell have fallen onto the gene in question. Then what we did last time is we plotted here the row means of this fraction on the x-axis. On the y-axis, we plotted this factor here, the ratio of row variances to row means, which is something like the excess over Poisson noise, because the Poisson noise, where we expect that the variance of each gene, so row var just means the variance of a gene taken over all the cells, and under Poisson conditions, so if a gene actually doesn't vary from cell to cell, then we expect to see a technical noise due to the Poisson statistic, which should be somehow proportional to the mean. So I divide by this, and then I see that the majority is some more or less constant, and the biologically interesting genes stick out from this. And these are and I took the top 2,000 of these genes and stored them here in this list of highly variable genes, as I called them. Again, what we did last time, we multiply these fractions with some arbitrary scaling factors and then add one pseudo count. This was this log1 plus function in order to get expression values. And I use this log1 plus in order to make sure that what used to be a zero count is now still a zero count, so that we are still in a sparse matrix. And now we go to the PCA. And I calculate the first 20 PCA components of this expression matrix. And in order to make sure that this takes only a minute and not an hour, I take only these top 2,000 genes, which actually vary from cell to cell. And this is why it was the first thing where I wanted to do a bit of theory and talk about why it's actually the PCA that we are doing here, and what precisely do we want to achieve with this. And to this end, I will come back to this in a moment, but let's first go through this, through the rest of the analysis to see what we are doing here. You remember our sort of rough explanation that we did last time was that we said in the PCA we reduce this expression of 2,000 genes just down to 20 principal components, and each principal component is now a linear combination of the expression values of many thousands of genes. And typically the principal component will take together those expression values, will take together those genes which are correlated with each other, so that the Poisson noise will average out and the signal will increase. So the idea is that in reality, our actual state space of a cell has a much smaller dimensionality, maybe only 20 or 30 dimensions, and each gene is influenced by several of these 20 or 30 state variables. And for now, we assume this influence to be linear, and we will later come back to this and realize that this is actually not a very beneficial assumption. But now, to keep it simple, we assume each gene is a linear combination of these state variables, and independent, and the other way around, we can say each state variable can be estimated by taking an average over all those genes which are highly dependent on this state variable. And then, because we are averaging over so many genes, we average out the individual Poisson noise, which is independently acting on each gene, and hence improving this signal-to-noise ratio of the actual biological signal versus the Poisson noise. And that's why we said we now get a much clearer structure and see much stronger differences between cells which are actually similar to each other versus cells which are very distinct from each other. And to see this, I now calculate a distance matrix by just calculating the distance of for each gene. Remember, each cell is now described by a 20-dimensional vector, and we just take the Euclidean distance of each of these vectors against each other vector. We have something like 10,000 cells, so this is now a matrix of 10,000 by 10,000, so 100 millions. That just barely fits into my computer's memory, but still it works. And if we have a look at this, 110, 1 to 10, you can see that we can now read off the distance of every cell to every other cell. And this distance now has some contrast. This cell, for example, is very similar to that cell and that cell and that cell, and quite dissimilar to these ones. In this contrast, where you really see groups of, where for a given cell you find cells which are quite similar to them and cells which are very dissimilar to them, that's something we wouldn't have gotten if we hadn't made the PCA. And if instead we had calculated our distances, not on the vector of 20 PCA coordinates, but on the vector of 10,000 log normalized genes. Because then most of the signal in there, which affects the distances, is simply noise due to this Poisson noise, which individually affects independently each gene separately, and hence it just gives you a huge amount of, it washes out all the actual signal and drowns it. So the next thing I can now do is I take a random cell. Let's take the cell 135 and ask, what is the distance of this cell to all the other cells? And if I do this and order it and sort it like this, then I get here at the beginning of this thing the cells top 20 nearest neighbors. And this is a useful thing to understand these data because we still have this 20-dimensional space, which is hard to visualize and hard to work with. And one way to get the topology of this space and understand what's happening there is to look at the neighbor graph. We will construct a graph where each cell is a vertex and each cell is connected to its 20 nearest neighbors by an edge. And now I have to find these 20 nearest neighbors. So I take my distance matrix, which here tells me for my chosen cell the distance to all other cells. And if I sort this, I find here the 20 closest distances. The first one is, of course, 0, the cell to itself. And then comes, this seems to be the closest neighbor. And then they go slowly a bit further out. Now, one thing you can do at more liberty is how do you now actually get the indices of these cells? In R, you do this by replacing the word sort with the word order. And what this now tells us is the index of the closest cell is 135. You remember I've picked cell number 135. So its closest neighbor is, of course, the cell itself with distance 0. The next cell, this one has index 405. The third closest neighbor is cell number 649, and so on. So this here is the 20 nearest neighbors of cell 127. And of course, I can make this logic that I've just done for all cells, like I've done here, by making a loop For the people who are not that familiar with R, just let's go through this again. We define here an anonymous function which takes a cell index and does this operation we've just done, namely sorting and getting the indices of the 20 closest values. And we call this function for all the values from 1 to the number of cells. And each time we get a list of 20 values, and then this sapply does this and puts them all together in one big rectangular matrix. If I now do this, it again takes a while. And takes another while, and then I get my nearest neighbor information as a matrix with 20 columns and 10,000 something rows. Each row is a cell, and the columns are the cell's 20 nearest neighbors. Yeah.
What metric are we using?
The metric we've used this here when we did the distance, that was the Euclidean distance on the PCA space. Now I have to wait a moment for my computer, so so now here we have this nearest neighbor matrix, hat NN, and you see how this looks like for the cell number 3. We see here the cell's nearest neighbors is the cell 3 itself and cell 10,073, etc. and so on. Just because you asked about what metric we are using, it's always a good idea to double check something like this. So let's look in this thing here and maybe look specifically at one of these matrices. Let's say distance of cell 23 to cell 45 is this. So in our PCA matrix, cell 23 is this here. Cell PCA $X, cell 45 is this here. If I subtract them, this is the difference vector between the two. I square the differences, sum them up, take the square root to calculate my Euclidean distance, and compare again with what was in my distance matrix, and it's the same. Which is a useful thing to always check whenever you use a function like this disk. Never trust R to actually really do what you think it does, not because R might do a mistake, but you might misunderstand the way how the function expects its input or what's in the rows and what's in the columns. But this time it works because I've triple checked it before already. So that's what we now have. You've just seen this took quite a while to find these nearest neighbors. And that you can imagine that this finding nearest neighbors scales quite badly. If you think about it, here we have a scaling with n, with a number of cells, because I have to go through this number of cells. Then, first to calculate the distance matrix, I have to go calculate the distance of every cell to every other one. That's a quadratic scaling in the number of cells. Here I have to then sort each row of this distance matrix. That's again an n log n scaling, because sorting algorithms typically scale with n log n. So this is slow, especially when we make n big, when we don't want to calculate this big distance matrix of everything against everything. It might not even fit into the memory, and it will take hours. Here's another function. There is a package called FNN for fast nearest neighbors. And if I call this, it just takes a short moment, and you see it already has these things. I just gave it my PCA matrix that I want 20 nearest neighbors, and let's compare what this thing gave. This is what we have. And let me make this a bit more space. And again, I want to be sure that my nearest neighbor thing has done the same. And the nearest neighbor thing, it gives me actually two matrices. One contains the index, and as you can see, it has precisely the same nearest neighbors. For cell 1, it's these ones here. For cell 2, 10,169, 10,169, it only has opted to not report the trivial neighbor of the cell being its own neighbor. Also, this thing here has a second thing, where it actually gives us a matrix of the actual distances to these things. which we had calculated, which we could, of course, read out of our distance matrix, but here we didn't. So in practice, of course, you want to use this thing. And this is a typical case, and this is a very nice example of something which, where what we've done here is a perfect way, is a perfectly reasonable way of getting started, just calculating a brute force, calculate the distance of every vector to every other, sort the distances, find the 20 smallest values. But once you want to do it often, it's worth looking up whether somebody had a better idea. In that case, the better idea that everybody had is the KD tree algorithm, which I can show you here how this works. Imagine here, we want to assign all your cells in a tree. And here we have a 3-dimensional space. We have a 20-dimensional PCA space. But of course, for the example, Wikipedia uses a 3-dimensional space. And you can imagine you make a binary splitting of this space. You first divide, if this were a space, I first divide it with a hyperplane here in the middle, and say, this is the left half of a tree, this is the right half of a tree. Then within the right half, I might make a hyperplane with here and say, this left half, you guys are the right half. And then I might make a third thing here, where our upper half is empty of people and lower half has something. Then for the fourth division in my tree, again split like this, then like this, then like this. And they've illustrated it here with this example, where they say we have some employees which are always specified by the 3 dimensions, age, name, age, and salary. The name is, of course, also a continuous thing because you can sort it alphabetically. And if you now have a certain person, then you can look, does the name come before or after Kevin in the alphabet? If it comes before Kevin, then you say, is the age greater or smaller than 60? And then you ask the third question, is the salary greater or smaller than 50K? And then again, you come to the first dimension, does the name come before or after Bob? And in this way, you sort all these things. This is maybe not the most useful example to do that, specifically because you're unlikely to have a query where you ask, given this employee, what is another employee which is as similar as possible in salary, age, and first letter of the first name? But this is, of course, what we do in a nearest neighbor search. We have a cell, and we quickly want to find its nearest neighbors. And for this, the this algorithm that I've just called here, when I call this FNN, arranges my cells in such a tree. And when it now has a cell, and I've just seen before that Wikipedia has a beautiful animation, which I can quickly show you here. So here they imagine we have a 2-dimensional plot with a number of points, and one point which will turn up here in a moment is as a red point, this one here, and we want to find the nearest neighbor of this red point. And as you can see, it now always makes here, it has arranged all the cells in this tree, and so it can always expand or remove the different things. So at the moment, what it has just done is that it said, okay, first let's look in the same 3/4 than my point itself, and we find something. Then we can ask, is there maybe a closer one here in that one? And of course we say, no, that's not possible because everything in here has to be further away from that one. But of course it could still be a closer one in this square here. And so it goes through all the squares, always expanding or staying within the area and ruling out whole blocks of a space, saying, okay, let's rule out this whole quarter, this whole eighth or something like this. So most of you might know the 1-dimensional case, the binary search. Who knows? Does everybody know how a binary search works? Yes, right? You can also say no, that's perfectly fine. If you have no, because actually you do know, if you have a phone book or something like this, you look for something, you open it in the middle, when you check, you have to look before, right? Now you again open it in the middle of the remaining part. In this, you always rule out whole halves of your search space. And that's what our nearest neighbor algorithm here internally does, and why it was able to give us a result within 5 seconds instead of, as we did before, within 2 minutes. Even this turns out to be slow for really big point clouds. So there is also areas like the approximate nearest neighbor search, which is if you have really billions of points, you want to very quickly find a neighbor, and you're happy even if it's not the very nearest neighbor, but just one of the nearest neighbors. And that's, I think I mentioned this last time, the library that everybody uses for this purpose was originally programmed by people from Spotify who probably used it to make their music recommendation. So this is how far we got last time. Now the next thing is I make a UMAP, and for now I simply call the UMAP function. So you see what the UMAP function does, it gets this PCA matrix, but in reality it doesn't really use the coordinates, it's only interested in the distance matrix. I could also have passed him the distance matrix that we've already calculated, but because the distance matrix is much larger than the PCA matrix, it actually becomes slower. But it's important to know that this is in principle possible, because later we will talk about what UMAP really does. So that's what I wanted to do next week. I don't think we'll get there this week. But one point that you can already now remember is UMAP, of all the information that humans uses, it only uses the information of which genes are of the distances from one cell to the next. It doesn't, it ignores completely everything you tell it about directions. It only uses the distances. So whenever it looks at a cell, it only calculates its distances between cells, but it never looks at individual coordinates. And the first thing it does with these distances is to calculate the nearest neighbors. And because we've already done that, I handed this over here and told it, the nearest, you can tell UMAP which nearest neighbor calculation method it should use. By default, it uses with FNN matrix here, but I tell it, don't use the FNN matrix. I've already done that. Here's the result. So now what UMAP gives you out is simply a big matrix of with 2 columns for the 2 coordinates and many rows, one for each cell. I put some row names on these 2 columns because I will need this later. Now it has U1 and U2. And now I can plot this simply, these 2 coordinates. And here you see now our data this time. We have here, and that's why I chose this data set, because now we not only have these blobs, but we also have lengthy trajectories. And what's going on here, as we will later find, is, let me quickly make sure I don't tell it the wrong way around. Yes. What's going on here is these are the stem cells, and these are the neuroblasts, which are about to go on their way to the olfactory bulbs. And so the stem cells sit here, they wait to be needed when they activate, and then they rapidly change state. A lot of genes change expression until they enter the cell cycle. And here they make a couple of cell divisions, and then they become neuroblasts. And this trajectory is interesting because, remember, we are looking at a steady state process. So there's continuously stem cells getting activated, dividing, and turning into neuroblasts. And because we just took one sample, we take a cross-section through these samples, and we see all the intermediate stages that a cell can be in on its journey from being a stem cell towards being a new neuron. And physicists will consider this as an example of ergodicity in a way, because what we here have is, you see an ensemble average here. We look at all these cells and we see many different, cells in many different states. But what we are actually hoping for is that we can retrace the the life journey of one cell, that we could really imagine a typical cell starts somewhere here, goes through all these intermediate states and ends up here. And in that case, this is both, we hope to interpret this as a picture of a given cell's temporal evolution, even though we know that actually it's just many different cells of which we have made an average of whatever state we called them in. And here we will typically simply assume that this is reasonable, and it sounds quite reasonable to say that we see all these stages, we see all these individual stages. But of course, it only makes sense if we assume that the system is in steady state. That it wouldn't matter if we take the sample now or maybe a day later or another day later, because it's always in the same flux. In thermodynamics, you might know this as the principle of ergodicity, where you always ask, is it appropriate to assume that your ensemble average is the same as the time average over one particle's trajectory? Here we take this for granted, simply because this is one mouse, which doesn't really change much. Of course, it is not really true, because we know that the mouse changes with age, but hopefully the change is slow enough that we can see this here. We can now, of course, take here our UMAP and add some expression of specific genes. Here, for example, this gene, double cortin, which DCX, which marks neuroblasts, which I have marked here just to remember myself which end here are the neuroblasts. I have a right end, or another one which marks the stem cells, namely the one which makes this glass protein that I mentioned at the beginning. No, 1A3. And as you can see here, you can see how this protein, how this gene goes down and the other gene goes up. For the biologists, it also take another gene that you might know well, MKI67, the standard marker that's always used to find cells which are currently in the middle of a cell cycle in mitosis and are presently dividing, and that shows us that here the cell division is happening in this thing. I can't quite remember whether I've already shown you this sleepwalk tool that we developed and always like to use here. But in any case, I show it once more. It's a little tool which works as following. I put my mouse on some cell, and it now colors each cell, each other cell, by its distance to the cell under the mouse cursor. And by distance, I mean Euclidean distance in the PCA space. So you see here, the colors is the Euclidean distance in PCA space under this thing. And this allows us now to see which distortions our UMAP has done. For example, if I sit here, I can see all these cells here. They are probably very similar, even though they are spread out over a rather large area. While this little thing here, there's a lot of change happening over this short amount of space here. And the same here, we see how strong the change is. Here again, we see that there's a lot of difference between here and here, much more than there's difference between here and here, for example. And these things, they are quite different and quite far from the other stuff. Of course, I like this tool because it's something we made. But apart from pride in one's own work, I think the value is it allows you to see how well this two-dimensional rendition has worked, because obviously it can't be perfect. We are trying to project a 15-dimensional space down to 2D in some nonlinear manner, and that must cause distortions. And with this manner here, I can somehow judge the distortions a little bit. So now, the other thing that I wanted to finish, but then do in a bit more, but we also need some more idea with this is, let me quickly show this. First, the plot, what we're heading at. it is nice if we want to reason about these things and do statistics, even though this is such a lengthy thing. As a first step, it is quite helpful to group the cells into groups of similar cells. And this is what I've done here. All the cells of the same color are close to each other in terms of neighborhood or in terms of distance. And this is helpful even though the division line is probably rather arbitrary, as you can see here. And in biology, it's often called clustering, this process, and each of these things is a cluster. In mathematics, we would probably not use the term cluster, because there a cluster means we have a point cloud, lots of points in our 15-dimensional space, and then there's one area where the points are much denser than elsewhere, and this is what we typically would call a cluster. Here, as you can see, every point was assigned to one color. It's not that we found clusters of big things and interstitial part where there's nothing, but nevertheless, the terminology has now stuck where people call this here clusters. Maybe it would be better, and if you look in mathematical literature, they like to call it communities or groups or something like this. And we'll come back to this, whether maybe using the actual cluster terminology as it's used in geometry or in statistics might be useful here. But for now, how do we do this? How do we quickly assign each cell to such a place? And there's a couple of different algorithms how to do that. Classical ones might be the k-means clustering. K-means that many of you might know, and there is this beautiful animation here in the in Wikipedia, where you have here this Mickey Mouse data set, as we call it, with one big blob at the bottom and one at the top right and one at the top left. And we would like to assign each point to each data point to one of these three colors in such a manner that they get close to each other. And here with classical k-means algorithm, which is sort of the archetypical clustering algorithm, which is maybe the oldest in high-dimensional data analysis, is we have three means, which are these small little crosses that you can see here. And you always make an iteration in two steps. In the first step, at the beginning, you just choose these three means completely arbitrary. Just put three crosses on this thing and say, these are my three means. And then I assign each point to whatever cross it is closest to. And once I've done that, I re-go and calculate over all the cells which I have been assigned to one of the three groups, the new centroid, so the new average of the x and y coordinates to get a new center. And that's what's happening here. Every time the thing moves, a new center was calculated. When the cross moves, when we reassign the points to what cross is closest to them now, when we again average over the points which these crosses which are chosen by these crosses. And in that manner, we get this assignment that in the end captures the structure quite well. But we would actually probably prefer the final result to not cut here, but to cut here, to cut the head from the ears of the Mickey Mouse. And to get this, the next thing that people like do, and that many of you might already know from other areas, is the EM algorithm, which tries to fit a multinomial Gaussian, tries to fit a Gaussian to each of them, and then notices that, well, in order to get the best Gaussian, I make one Gaussian with mean here, and with mean here, and the yellow Gaussian gets a wider standard deviation than the blue and the red Gaussian. And in that way, we also then iterate in a similar manner. But for our single cell sequencing, all these algorithms have a bit an issue because they assume that there is a certain density of points which we say, okay, these points belong together if they are that dense. So that your typical data looks like this here. But in single cell, what we often have is that we have for some cell types, they are very dense, and another cell type might look like this here. with much wider space. So what we consider dense enough that they should be clustered together is very different in different areas of our 15-dimensional PCA space. In some areas, we might consider this here as dense, and this here even as, in this distance, as enough to say these are two different clusters, and here this is just enough to say which cells are in the same cluster. So we would like to somehow to normalize for local density. And once you think about this, you realize, well, one way to find is to find out the local density, and to do that, we might want to connect each point to its 20 nearest neighbors and just ask how far away is the 20th nearest neighbors, and that tells us the local density in this point. And then we might take this as a density, and so we normalize for the local density by not asking what's the absolute distance, but how close are they compared to whatever is the nearest neighbor, the typical closeness of nearest neighbors here. And once you get there, you start forgetting about the absolute distances, and you only care about relative distances. Distances relative to what is typically in this area in the PCA space, the typical distance to the nearest neighbor. So you say 2 points should be clustered together in the same cluster if their distance is similar to the distance to nearest neighbors, otherwise not. Which in other ways says we want to cluster points together which tend to be each other's nearest neighbors. And a cluster should be a group of cells which are all nearest neighbors to each other, or all have a large overlap in their nearest neighbor. And that's what we have used here in this Leiden clustering, where we have made an assignment such that each point is a nearest, such that points which have a huge overlap in their neighborhood tend to get the same color. And in order to that's now so that we finally do a bit of math. I thought this was one of the two things I wanted to show you now for today in terms of concrete mathematical aspects. Actually, I wanted to start with the other one, with the PCA, and explain why exactly we use the PCA, but now I've just started talking about this, so I continue with that side. What we now want to do here is we want to, we first look at this neighborhood graph. That's what we've just constructed. You remember we used this FNN function to find for each cell its 20 nearest neighbors. And these 20, and what we now do is we define a graph. As you probably all know, just to make it clear for the non-mathematicians, to make the terminology clear. In math, the term graph means an entity where we have vertices and edges. The vertices are objects, which we just number, and here our cells will be the vertices, and the edges are connection between cells. So a graph is just a manner of taking a list of entities, such as cells, and then defining how these things are related by either drawing a line connecting two things or not drawing a line. And these lines we call edges. And what we've now constructed before is a neighborhood graph, where we connect, where we say each cell is a vertex and each cell is connected with its 20 nearest neighbors. And there is a software package which is generally used for all this graph theoretic calculation. It's called iGraph. It's actually a C library, and there is interfaces to use it from Python and from R and from many other programming languages. And we use this now here, and I will construct now the graph. And to do this, where do I have to start for this? Yeah, I load my iGraph package. And now to show you how this iGraph package works, or maybe what I have to graph. So usually when people think about graph theory, they typically think about this. This is a graph with 6 edges, with 6 vertices and 7 edges. And if you want to describe it, important is only the connections count. The fact where the vertices are on the screen doesn't matter. If you want to describe a graph completely, all you have to do is to make a list of edges, saying my graph has vertices numbered from 1 to 6, and the connected vertices are 1 with 5, 1 with 2, 5 with 2, and so on. You give a list of vertices. And two graphs which have the same vertex list are considered the same graph, even if I draw them completely differently by putting maybe rearranging how the vertices are on there. And now let's construct this graph. So let's take, and for this I have to make an edge list which connects each cell with its 20 nearest neighbors. So let's start with connecting each cell with its third nearest neighbor, k is 3, and then we will connect it also with the nearest, the second nearest, the third, the fourth, and so on. The third nearest neighbor, what I have to do is I look in my nearest neighbor thing at the column 4, because you remember had n. These here, this column is the third nearest neighbor. So I now have to make a list of connecting every graph, so every of these, with every third nearest neighbor. And here you see how this looks like. Vertex 1 should be connected with vertex 2, 2, 2, 2, vertex 2 with 7, 2, 5, 7, and so on, vertex 20 with this, and then this goes on to vertex 10,000. And if I now let k run through 1 to 19, then I get everything from the nearest neighbor to the 19th nearest neighbor. This iGraph package is a bit peculiar because if I want to give it its vertex list, it doesn't want it in this thing, but it always wants it as a list of numbers. First edge start and second edge start, second edge, third edge start, third edge end, and so on. So this is why I have to take this thing and now have to read it off like column-wise, going from here to here to here to here to make this long vector. And I do this for all the vectors. And when I get these long edge list, which is a really long list with 600,000 edges, and for all these edges, I now get and now I can tell it make a graph by storing all these edges. And this I now call my NN neighborhood graph. And it connects each cell with its nearest neighbors here. And I can now ask a couple of graph theoretic questions. One, for example, would be the degree of a vertex. I ask each cell, how many edges do you have? With how many other cells are you connected? And as you can see, most cells are connected with more than 20 nearest neighbors. And this is because we made an undirected graph. You might see in here that I said directed is false. That means often two cells don't agree. Cell 1 considers cell 2 as one of its nearest neighbors, but cell 2 doesn't consider cell 1 as one of its nearest neighbors. For example, in a situation like this, where we have 3 cells which form here a group of people who are happy with each other and who consider each other their neighbors, and now we have an outsider here who considers these 2 cells its nearest neighbors because there's nothing else nearby, while these 2 cells doesn't consider that one as one of its 2 nearest neighbors. And for now, and this will bite us later, and we will notice that this causes quite a bit of issues, but it's still the simplest way of going it, so people go with that, but it's important to keep this in mind that we have symmetrized our graph. Originally, we could say the graph is directed. We can say this is a neighborhood relation. This cell considers that a nearest neighbor, but not in the opposite direction, while these edges are bidirectional. But we didn't consider that. We removed the arrows. We say the graph is not directed, and now suddenly this cell here has more nearest neighbors than this cell. If I make a histogram of these graphs, you can see that most of the cells have something around 35 neighbors. And quite a few cells are really popular. They have 100 neighbors, even for, so we have way more cells which consider them neighbors than they consider neighbors, like these down here. These are probably the cells which sit in the center. If you have, imagine you have a cluster which sort of to the outside gets slowly more diluted, then the cell in the inside will be one of these here. So, And now there is a functionality called the Leiden clustering. And if I run this Leiden clustering, it's an algorithm which is in this nearest neighbor, in this iGraph package. It assigns to each cell a number, which we call its community membership or cluster membership. As I said, in biology, we call them cluster. In the graph theorists prefer to say community because they want to reserve the word cluster for things which are really tighter than the rest. So, and this is what I've shown you here in this picture, where I added for each cluster, where I used my ViewMap, and then I have here this result, what this cluster membership has given me, this Leiden function. What the Leiden function does, it gives me a vector which just tells for each cell which cluster it has been assigned to, so which color it should get here, and that's what I do here. I do this, and I look at all these clusters and say cluster 1, 2, 3, 4, 5, 3, 3, and now I color them, and I see them. And now you might ask, what is the criterion by which this Leiden algorithm has assigned the clusters? To explain this, we need a little geographic hack, because there's two algorithms which both start with L and are slightly different. They are called the Louvain algorithm and the Leiden algorithm. Louvain is the French name for Leuven, which is in Belgium, and Leiden is in the Netherlands. And the people, somebody at the math department in Louvain or in Leuven has suggested a criterion how one could make such an assignment of clusters, and suggested an algorithm, and then other people in Leiden, 50 kilometers over the border, suggested a good improvement to the algorithms of their Leuven colleagues. This is now we always talk about the Leuven score and the Leiden algorithm. So I first want to explain to you the Leuven score and skip over the improvement that the people in Leiden did. So what the people in Leuven, they were not concerned with single-cell sequencing. This whole stuff is older. They were concerned with big graphs and wanted to find modules in the graph. So the idea was, if you have a graph like this, you might say, this here is a module of clearly connected things, and this is a module of clearly connected stuff. And this graph somehow falls into two modules or two groups, this vertex group and that vertex group. And if you now give them a graph, how can you find in a mathematical, stringent, clearly defined way such groups or communities of vertices which are much more closer connected to each other than all the other ones? And that's what we do here on our neighborhood graph. And what they suggested to do here is that they first define a score. So we do it in a bit a backwards way, rather than coming up with an algorithm how to find something like this. They say, let's assume we already have an assignment of neighborhoods. How can we score how good it is? So let's assume somebody comes up and says, these are my one community. Should see if I find the second color. This here is the second community, and that one is the third community. How can we give a score on how good a clustering this is? Because obviously it's not a good clustering. We can make it much nicer by putting the green one, the purple one here and putting the green one over here, and we might debate whether it's really a good idea to have 3 colors. Maybe we should only have 2 colors. And so to do this, they decided to count the number of edges that stay within a cluster. So these are within cluster edges. They are I call this a within cluster edge or within group edge because it connects 2 vertices of the same color. And in this clustering, we now see that we have 1, 2, 3, 4, 5, 6, 7, 8 within edges of 9, 10, 11, 12, 13, 14, 15, 16. 8 of 16 of the edges are connecting two of the same colors, so are not sort of breaking the circle or the community. So we might say the proportion of connection is 50%. 50% of the edges stay within color, 8 from 16. And is this a lot or not, 50%? That was now the issue where they wanted to score this. We have to compare this with what we would expect among randomly. And to do this, they came up with the idea that they said, let's replace this graph with a random graph and see what the percentage of within community edges would be then. And then we can see how much we exceed this random stuff, so how much our assignment really have brought the community together rather than just assigning colors at random. And whenever you do something like this, you have to decide on a randomization scheme. You have to decide, what do I mean by assigning this at random? Do I want to assign the colors at random? So for example, I could now say I have 3 purple dots, 4 green dots, and 4 red dots, and I just shuffle the assignment of memberships to points. But if you think about this, this is not quite ideal, because if you do that, you will quickly come to a conclusion that the obvious thing is to assign only one color. No, actually I've forgotten the exact reason why. In the end, they came to the conclusion they should randomize not the assignment to the edges, but they should randomize not the assignment to the vertices, but they should randomize the edges. They said, if the edges were randomly connected, would we still get as much modularity, so as much within color connections as otherwise? And to do this, they decided to define randomly connection as follows. They break each edge in half. In these two halves, they call the two edge stubs. Each edge has now 2 stubs. In each graph has also different stubs. This is a graph, this is a vertex with degree 4, so it has 4 stubs. And now we reconnect the stubs by picking each stub and finding it's a random other stub as partner, and randomly connect the stubs to each other and see how much vertex coloring, how many of the stubs we then find which end up connecting to a stub of our own color and not to a different color. And then we compare this and see the difference. And this is one of these things where, when I read this first in textbook, when I read the Wikipedia page, I found it really hard to wrap my head around it. Why is that good? Why does that help? And then I noticed, actually, it's so hard to understand because it's so hard to describe, because we try to describe here something in mathematical terms that probably the people came up with by actually programming it, because once I sat down and tried to write it in R, it suddenly sounded pretty obvious. And I want to demonstrate this to you now. So what we want to do here is what our Louvain algorithm has done is it has found an assignment of points to colors which makes the score that we want to calculate very high. And where it has reached a modularity score of 0.84. And in order to understand what this means and why this is high, we try to find out what this modularity function here is actually calculated. So let me show you how this looks like. And to do this, I take my graph and say, turn this graph into a data frame. Now you see here I have a data frame which tells me for each graph, for each edge, its vertices. So I have one vertex from cell 1 to cell 2, 6, 9, 7, one from cell 2 to cell 10, and so on. These are all the vertices. And what I now do is I look up for each of these vertices which color it has. Here. now you see, I see the first edge which is in here connects color 1 to color 1. The second edge connects color 2 to color 2. And here is now an edge which breaks color. It connects color 5 to color 2. So I can now easily calculate how many of them connect same color and how many connect different colors by just asking which percentage of these rows here have the same value here in this column and in that column. And I do that by just using summarize and asking what's the percentage of rows where from cluster is equal to cluster, and I find in 92.3% where edges connect two colors of the same cluster. That sounds high. It seems that the Louvain algorithm has found an assignment of colors to vertices which really made sure that nearly all edges, or at least 92.6%, stay within the color. But if we now did this permutation of randomly reconnecting each edge with another edge, how does this now change? And so what I now do to simulate this, I again take my matrix here and use the same code as I did before, but I add something. Namely, here I take the whole column 2, so the second of these two columns, and permute it. Just randomly shuffle it order, so that each vertex here has to find a new vertex to connect to. And this is what the sample does. In our sample means permute the order of the elements to a random new order. And if I do that, and I get now the same as permuted, and now I again do what I've done before. I look up for each cell which cluster it is in, and then I calculate which percentage of all these edges here connect two vertices of the same cluster, and find it's only 8%. What we now have done here is notice that this is exactly what we've done here. And now it's also clear why this edge stub thing is such a strange permutation scheme. If I describe it like here, saying we break the edges, and when every edge does something new, this sounds a bit arbitrary. But once I programmed it, I realized, yeah, of course, we just took the list of all edges and just permuted one of the two columns. That's what we've done. And what they then argue why this is a beneficial thing to do is that they say it preserves the vertices. Because in order to have a fair comparison of how many vertices, how many edges you would expect to connect vertices of the same color, you should look at random permutation where all the vertices contain their color, because otherwise it would be unfair. A vertex which has a high degree has to work much harder to be only connected to its own color than a vertex with a low degree. So I want to preserve the number of the vertex degree. And this is why they chose this permutation scheme, because it comes up with a new graph where every vertex still has the same degree as before, but completely randomized connections. Here I used a random sample, so I made a random permutation. This is why I should execute this code here a couple of times to see what values I get. 8.1%, 8.2%, 8.2, 8.3, 8.1, 8.1, and so on. On average, 7.7%, strange. Just an hour ago, it was always 7.9%, not 8.2%, which is a bit annoying now, but let's ignore that. And if I now subtract these two, then I get the modularity score that this thing has calculated up there. So, and now we have now seen why this modularity score, what it actually measures, and why it makes sense. And as promised, it has in a way nothing to do with biology. This is an idea which people came up, I think it came up researching the solid sea looking at big Facebook connection graphs or something like that. At least if I remember right, this was the example that they used in their paper. And this is now how we And the question would now be, how do we find an assignment which maximizes this modularity? And I want to give you an idea of how the Leiden algorithm works and why it's actually a simple Leiden algorithm. And this will also tell you why this modularity score is so popular, because it's so easy to calculate. And it's easy to, because it's easy to calculate, it's easy to optimize it. And we'll see in a moment why. But at the moment, it might look to you that it's not easy to calculate. I had to make a random permutation, and then I rerun my data once more. And every time I rerun it, I get a slightly different result. So how can I do this that I don't have to do this random permutation? I want to do this in a deterministic manner, not with a randomized algorithm. So what to do there? Well, what we now calculate is the following. So in order to calculate is the first thing that I do is I calculate for each, I make this table here, which you see here is the vertex. Here is the group, so the color of the vertex. So these are the cells, these are the colors of the cells. And here the middle one tells us the degree of a cell, so the number of vertices. If I now take all vertices of one group and add them all up, all the degrees of a vertex, then I get the total number of stubs in this cell. So let's do this here. I sum up all the degrees from the same group, and now notice within the group with color 1, there's 46,000 outgoing vertices, in the next is 36,000, and so on. So this I call the stub count, because it tells us how many stubs, how many outgoing vertices. And as you've seen, it's just the sum of the vertex degrees of all the vertices in a given group or color. And now, if we want to, if I now want to know, where have I put this now? At stubs or something. Where is my note? So, and now I want to calculate the modularity score. And first, I wonder if I now have these stubs here, these stub counts, how many edges are there in total? Very simple, I just add this here up. Stub counts $n. This is the number of stubs, and if I sum this up, then I get the number of edges. Actually, I get twice the number of edges, because for each edge appears twice here in this table. Once is the first, once is with its start vertex, and once with its end vertex. Now I have 347,000 edges, which is what I had before, and I ask how many of these edges are connections between vertices within each stub. And let's see, and for this, I now have to say, wait, some stub counts 0.77, 0.08. This is from above. Stub counts. Let me skip ahead to the end, and then we make this with a matrix, because then it makes more sense. Yes, that was. So to do this, I now turn the whole thing into I make a stub matrix. I make the following matrix. First show you how it looks like, and then I explain how I made it. This is my matrix, which just tells me all the connections from each group to every other group. It tells me there is 12 edges connecting a cell in vertex 1 with a cell in vertex 3. And 1,200 connections between cluster 2 and cluster 5. And of course, there's 22,000 connections within cluster 1, 13,000 within cluster 2, and so on. In order to get this matrix, I've just done the following. I took again my edge list. added in the edge list again with information of what a cluster these two things belong to. And now I group by from into cluster and make this table here, which tells me from cluster 1 to cluster 1, there's 22,000 connections. From cluster 1 to cluster 2, there's 2. From 1 to 3, there's 12. And then I use this little bit of, and then I just turn this thing into a matrix, because now it's below each other, and I want to have it as a matrix like this one here, where I can read off from and to on these two directions here. And because, of course, every edge either appears on one side of a diagonal or on the other side, so it can either appear here. Here are the connections from 1 to 3, and here are the connections from 3 to 1. But of course, I want both connections. So I add the transpose, and now I get a symmetric matrix. And in the symmetric matrix, I now have here the same value on top and bottom. So each edge now appears twice, once on the top and once at the bottom. And now I can ask which percent of all the edges are connections from a vertex from a vertex in one group to a vertex in the same group. And that's the diagonal of my matrix. Everything in the diagonal are connections within a vertex. And everything outside the diagonal are connections from one group to another group. So this here are all the connections within cluster 1, 2, 3, 4. And if I sum them up, then I get all the connections that stay, all the edges that stay within a cluster. Actually, twice as much, because I have counted everything doubled. And this is the sum of all connections I have in total. And if I divide it, I see 92% of all the connections are connections that stay within the same cluster, because 92% of all the edge counts sit in the diagonal of this matrix, which tells us how many edges are there from each cluster to each other cluster. And now I ask, what happens if I permute these edges? What I now do is I first go back and calculate my word and calculate the column sum. So I again ask for each group, how many vertices come out of this group? So what's the sum of the degrees of all the vertices in there? And now I can ask, how does this work? What is the probability that an edge goes from vertex 1 to vertex 1? The probability that it goes from vertex 1 to vertex 1 is of course this number multiplied by itself. If I take this and divide it by sum, by the total sum, then I see here, this is for, if I choose a random edge, this is the probability that the random edge I've chosen has started in cluster 6, and this is the probability that it has started in cluster 2 and in cluster 3. And if I take this thing and square it, then the following happens. This is now the square of what I had before, so this is the probability that the vertex, that an edge starts in group 1 and ends in group 1, because I have taken this probability to the square. This is that it starts in group 2 and ends in group 2. So this is the probability. So if I sum this up here, then I get the probability that if I take a random edge out of our permuted things, if I draw a random edge, that it ends in the same cluster where it starts. And this is the 0.8. And now with this, we can now divide, take this thing here, saying this is the probability that in a randomly permuted graph, a given edge connects two vertices of the same cluster. And that thing with the diagonal, what we had before, this is the fraction that we had before. And so the difference between two, that's the modularity score. And this is exactly what iGraph reports to us. And so now you've seen that our core element that we need is with CCM matrix. And now I can also tell you how the Leiden algorithm works, because what you've now seen from this formula here is once I give you this CCM matrix, this matrix which tells you how many connections are from each cluster to every other cluster, you can quickly calculate the modularity score. And now you might take one random cell and say, this cell is currently assigned to cluster 7. What if I put it to cluster 9? How will the modularity change? To do that, you just look at this cell and say, this cell is in cluster 7 and it has connections to cluster 2 and 3. So it contributes 1 to this count, 7 to 2, and 1 to this count, 7 to 3. If I remove it from cluster 7, I have to reduce these numbers accordingly in the table. And if I then put it to another cluster, then I have to add correspondingly. So you can easily change this matrix, and whenever you wonder what happened if I moved this vertex from the group it is currently in to another group, you only have to update according to this vertices outgoing edges the table to see how the table changes. And then you can look at the formula how the modularity score changes. So for any potential change, you can calculate very easily with just 2 or 3 additions how the modularity score changes if you move this thing to the other. And now you have a Leiden algorithm. What they suggested is start with assigning each cell to some random, cluster, and then go through all the cells in order. And whenever you take a cell, ask for this cell, does the modularity score increase if I move this cell to cluster 1, to cluster 2, to cluster 3, and you just find out which cluster do you have to move it to maximize it, to get the maximum modularity score. When you put it there, when you go to the next cell, again ask, in which cluster should this thing be in order to give the maximum modularity score? And as I've just said, this is easy to find out. If you have a cell, it really only requires 2 or 3 simple additions or subtractions to see whether the score goes up or down if you move the cell from one cluster to the other. And that way you can always go through all the cells one after the other in order to every time find a better modularity.
How do we decide how many clusters?
If I remember right, you start with having way too many clusters initially, and what then will happen is that quite often clusters vanish while you do this, because you have a cell which is sort of the only representative of its cluster, and you realize your modularity score goes up if you put this cell into a cluster with many cells, when this cluster vanishes. It might even be that the rule was at the beginning, every cell is its own little cluster of 1. And then once you go through, you do this. Now this algorithm that I've just described is what you call in computer science a greedy algorithm. A greedy algorithm is an optimization algorithm that tries to optimize some number, like here our modularity score, and does it in a short-sighted way. It always evaluates its current option and takes the one which currently evaluates it mostly. Greedy algorithms has the advantage that they are very easy to implement because you just do a loop, and they can be incredibly fast because usually for each step it's very easy to find out whether it improves or it decreases the thing. But the greedy algorithm has the disadvantage that it can get stuck in local optima. There's no guarantee that a greedy algorithm will find the best assignment with the highest modularity score. And that's where this improvement that I mentioned earlier comes in from the Louvain team over the idea of a Leiden team. They had a close look and they noticed that the Leiden algorithm tends to get stuck in one strange manner. It always finds a result where it has too many clusters, and where it might have 2 clusters where the modularity score would really go up if you put these 2 clusters together and made one big cluster out of these 2 clusters. But the Leiden algorithm can't see it because it always tries to move only one cluster to the next cell from one cluster to the other, which reduces it. But if you would take all the cells from one cluster and add it to the other, it goes up. So the Leiden algorithm is go from cell to cell, and for each cell always ask which cluster should you put it to maximize the improvement of a score, and go round and round and round till you don't find any improvement anymore. The Louvain algorithm then says, and once you reach the stage, now pretend that every cluster that you have is actually one big cell. And now always try the moves of taking all the cells in a cluster to a new one. So in a way, we take, once we have our optimal score, like say, this one here, it now says, okay, now make a new graph, which is just this green graph, this blue graph, this green graph, this red graph, with connections, with corresponding connections, and now run the Leiden algorithm again, but always moving this whole blob as a whole, not single cells, but move this whole thing to another one, or this whole thing to another one, until you find another optimum, where you probably have merged a couple of clusters, and then do this again, and reiterate until you're through. This is still a greedy algorithm, only a greedy algorithm which takes a step back and decides, okay, now we look at bigger moves, but we still do it in a greedy manner. But, and because of this, it's still terribly fast, and it can easily find a very good clustering, even if you have billions of points, and that's why it's very popular. Nobody has come up with a good idea to figure out how to make a non-greedy algorithm which really has a chance to step over local optima and find maybe a global optimum. Probably you can, as usual in these kinds of things, prove that it's probably NP complete and that you can't find the global optimum anyway. But in practice, it turns that usually the stuff is good enough. So that was now it was actually not my plan. I wanted to do it the other way around and tell you, yeah. The resolution parameter, yes. Kata has already worked with his software, so she knows that there is a resolution parameter. At the moment, for the actual modularity, we have defined our modularity at the moment as the fraction of edges within cluster, mean minus the expectation for this fraction under random edge permutations. And we try to optimize this modularity score. Now, and you will find a certain number of things here. And now there's something called a resolution parameter, and I show you first what it does by rerunning my clustering here. Where have I put it? Clustering, here. Here it is. So this is my normal clustering, and I can give it a resolution parameter, which has a default of 1. But I can, for example, put it to 2. And when I get much more clusters, or if I put it to 3, I get even more clusters. And if I put it to 0.2, then I get less clusters. Like here, I get only a few. And you can imagine that's useful to be able to adjust the number of clusters you get, because sometimes you want to have a more coarse-grained clustering, or sometimes you might want to have a more fine-grained clustering. And it turns out there's a very easy way to do that. You put a factor in here. And if you make a factor if you put a factor larger than 1, then this means that in order to optimize, it has to overemphasize this over that, or underemphasize, one has to think about it. But with some thinking about it, you will realize that this gamma actually causes the thing that an edge is not as much penalized or is more penalized for connecting two clusters of different things and then allowing to increase or decrease the cluster boundary. And it's also easy in the Leiden algorithm if we can calculate how the clustering changes, how the probability, how the score changes. And of course, changing this is simply a matter of putting the gamma in here. Because remember, this is how we calculate the modularity. We just have this matrix. We take the sum of the diagonal, the sum of the rows and columns, and add up these sums according to the schema here. And yeah, and if we then change an entry in the thing, then we can easily see how this goes up or down. So this would now be one fun exercise, could now of course be, now I have told you the formula. What I haven't told you is the update rule. The update rule means if you now know that in this CCM, in this matrix of this CCM matrix, the entry at position ij increases by 1 and at that position decreases by 1 because we have moved the vertex from here to here, how does the modularity score change? And you can try to figure out this update rule by yourself and then use this to implement the Leiden algorithm. I'm not sure yet whether this is fun, but maybe that's a fun exercise to actually for us to get a bit into actual programming. Let's try on Wednesday to actually implement the Leiden algorithm by first staring at this formula. And maybe we have to write down the formula with some notation with i's and j's and indices, but if we stare at it long enough, we should figure out the update rule, and that should then allow us to implement the Leiden algorithm. I haven't done it yet myself. I also won't. If we do it on Wednesday, then you help me with it, because I don't want to try to get confused by all this. Is this added or subtracted? Is this row or column? Yeah, this is something you better do in a big group, because then half of the people can think and the other half can check whether somebody has changed the indices. So let's try this as a little programming exercise, which, yeah, it doesn't sound very much like a biology exercise, more like a discrete mathematics exercise or concrete mathematics, which might, but let's see how this works. And the other thing that I wanted to explain to you more is we have to go back to the question of why the PCA is a good way of reducing the dimensions. And for this, I want to explain the PCA in the framework of, in a conceptual framework called autoencoders, which some of you might know well and others not, but we'll see. So, yep, let's go on from this point. Further questions? Okay, then, yeah, as you've seen this code here, I hope it's halfway explainable, but maybe it needs a bit of cleaning up. I don't know if I'll try to do that in the next days. Then see you for those who want to play a bit with programming themselves on Wednesday, otherwise on Monday.